जो है सो अब देखते हैं। नोट टेकर को तो रहने नहीं देगा ना पड़ेगा ना, तब तो सारा चीज ये तो फाइन। So good evening all the participants. As you all know, this is Science Dialogue series initiative of net preparation, you can say. So we have started with 4th subunit of net syllabus. This is 20 hours course, as you all know from flyer, and Dr. Ashwini Kumar will take all complete 20 hours lecture. And as you all know, Ashwini Kumar ji is PhD from NIT Raipur and then postdoc from IIT Delhi. As you all know, IIT Delhi is first ranker in this QS ranking. Recently QS जो है ना, QS ranking ने वर्ल्ड इंस्टीट्यूट की रैंकिंग की है, जिसमें इंडिया में फर्स्ट रैंकिंग आया IIT दिल्ली का. Am I right, Ashwini ji?
Yes, sir.
Yes. And nowadays Ashwini ji is teaching at Sharda University, Greater Noida. He has a long experience and he has also qualified several national level exams. That's why we have requested him, and he has given consent and is ready to deliver lecture and question answer also. Not only lecture, but you can discuss entire thing. And when I have, with the help of my student, when I have tried to find out the weightage of this unit based on previous year's questions, so this is a highly weighted unit, 4th unit. C part, especially C part major, means out of 13 unit, in C part of the question, Hannah, there are multiple questions, many questions are from, you can say 4th subunit. So don't ignore this, you have paid also, and don't ignore, attend all 20 lectures, so that possibilities of qualifying all those exams, including net exam, will increase. As well as if you are teaching, so you become better teacher after this 20 hours of lecture. So with this, I now invite Ashwini ji to continue. You all can join with this link which I have given. Same link will work every day. So Ashwini ji, welcome. And now you can start, you can show your schedule and other things. Meantime, I can request other participants. So after this, you can say 20 hours of lecture, first unit will also has been announced. So if you are interested and if you are satisfied with this, so you can join first unit course also, molecule and their introduction. Also other subunits teachers are in pipeline, so that either you can attend all subunit, 13 subunit or whatever we announce, or if you are interested in only few units, so you can attend that particular unit to improve. Because sometimes or most of the time we have observed that no need of learning all the 13 subunits or 14 subunits. If you are learning in depth only few subunits, so probability of passing or qualifying will increase. If you are learning 4-5 unit in depth, then you can qualify. Yes, Ashwini?
Yes, sir.
Yes. So I request you all to open your camera. आपका भी नहीं चेहरा दिख रहा सुनी जी साहब से.
सर आज जहाँ पर बैठे हुए सर, वहाँ पे ना मेन लाइट पीछे की तरफ है.
मेन लाइट पीछे है और आप वो भी कर दिया लग रहा है ना.
सर बैकग्राउंड हटा दें जैसा मुझे लगा इससे
चेहरा
थोड़ा सा
हाँ चेहरा थोड़ा सा साफ नहीं दिख रहा है.
सर कल से थोड़ा सा हम वो जगह चेंज करेंगे.
प्लीज कंटिन्यू.
Okay, so good evening everyone. So if you wish, you can switch on the camera. There is no mandate for that. So I would like to start with asking how many of you are actually preparing for CSIR UGC NET this time for July? You can raise your hand. Okay. Bitra. Okay. And Dhatri. Okay.
तनीषा नहीं. गुरप्रीत इज CA. वो आपसे न्यूरो वाले सब, कैंसर वाले सब वो डिस्कस करेंगे. Am I right? गुरप्रीत सिंह.
Yes, sir. Basically, I will talk later, but currently I would like to understand the in depth of
Yes, 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 yes.
cellular communication and cellular signaling. And I
Great.
think the time allow permit after the
Yes,
few
yes. After this, na,
minutes.
after every day, after lecture, you can connect, hai na? Because we have
Definitely,
so long
definitely.
questions, so many things, hai na? So
No,
first understand.
it's very
Yes.
interesting to connect with people. Gurpreet, are you from the this background, or you are just interested in understanding the things?
नहीं,
Yeah, so
आप कंटिन्यू करिए सुनी जी. He will ask question after
Sure, sir.
lecture, है ना?
Sure. Okay, sir. So those who have raised their hands can just undo the thing. So we'll just start with. I'm just switching off the camera so that, like, switching on the camera itself takes a lot of internet speed. So I'm just switching off the camera. I'll be on the mic, and I'm sharing the screen. Okay. Is my screen visible?
Yes, yes, visible.
yes, sir.
Okay, great. So, I'm Ashwini Kumar. I am presently working as assistant professor in Department of Life Sciences, Sharda School of Bioscience and Technology, Sharda University. It's located in Greater Noida, Uttar Pradesh. So, if any one of you wish to connect with me, on the left hand side you can find my LinkedIn, and on the right hand side you can find me on Twitter. Okay, so this is the syllabus of Unit-4 for CSIR-UGC-NET. On the left-hand side, you can see the current syllabus for this July session. And on the right-hand side, if you are preparing for NET, you might be knowing that starting December 2026, CSIR-UGC-NET and DBT-JRF is now merged. So there will be a single examination for CSIR-based labs and DBT-based labs. And the syllabus will be, syllabus is a bit changed, but fortunately, when I was going through the syllabus, I found Unit-4 almost same. There is hardly any change for the July session and for the December session. So in this 20 hours of class will be, for this particular unit, we'll be discussing about host parasite interaction, cell signaling, cellular communication, some basics about cancer. I'm saying basic about cancer because cancer is extremely vast subject. And the last unit that is given here is innate and adaptive immune system. But I would like to start from the section E, that is immune system, because unless until you understand immunity, you cannot understand cancer. Cancer is such a intricate thing. I'll be covering B and C together because they both are almost linked. So I'll start with E, then I'll go with B and C. Sorry, E and then A and then B, C together and then D. So I'll be covering the cancer section in the last. Okay, so I want to give this session extremely interactive. You can stop me at any time and ask whatever doubt you have, but I would like to start from a question, a throwing question for you all. How will you define immunity? Anyone, whatever is there in your mind, you can speak.
Sir, can I speak?
Yes, yes, definitely.
Sir, immunity is something if like intruder come, let's say India, Pakistan. If Pakistan intruder come inside our faculty, let's say inside the border, and
Mhm.
how our defense forces are dealing with such criteria, and such is the thing will follow with our immunity system. So
Okay,
in that, any pathogen, bacteria, microbial enter inside our body, and
okay,
how the body will respond. The first mechanism is something which is hydrochloric acid, which
okay,
generally kills most of the bacteria but before it is reaching to the duodenum and going. And I think that is the
okay.
that,
Sorry to interrupt. Sorry to interrupt because I made the presentation in a full screen mode. I'm not able to see the name of the person who is speaking. So before speaking, can you also mention your name? It would be easy for me to interact by calling
yeah.
name.
Sir,
Yeah.
my name is Gurupreet. Yeah.
Gurpreet. Okay, Gurpreet. Yeah,
My
yeah,
name is Gurpreet. So
yeah.
I would like to say if any, let's say Pakistan and India, and Pakistan send a missile to India, and if it reach within the territory of India, and how our India like defense forces are dealing with such a criteria. Similarly, in our immunity system, any pathogen, bacteria, microbials, and enter within our body, and how our body will react like within our system. The very first thing is the hydrochloric acid. As
Right.
soon we eat anything, food goes inside our body, that hydrochloric acid is so much acidic that it kills most of the bacteria. And now
Okay.
nowadays the our human like hydrochloric acid is not like highly acidic. That is the reason most of the bacteria are flowing to our body, and
Okay.
many pathogen are generating in our system.
Right. Okay. So I mentioned one analogy that if something comes from like something is foreign and it is attacking the body, then body resists the attack. Right.
Yes, sir.
Okay. Okay. What in case something from inside it attacking the body? Like there is nothing coming from outside. Then that is also very possible.
Yeah, so that is also possible if
Like
our
the analogy that you have used, I may if I may use, uh, uh, for very long there has been disturbances from internal groups also. There are a lot of internal groups that are trying to create problem, and a lot of people have been killed because of those. So that's a completely internal issue. Like that can also happen in the body. Nothing is coming from outside. Then how will you define immunity?
Yeah, let's say for example of free radicals, and free radicals
Okay.
are very highly reactive molecules that
Okay.
interact on the body and damaging the mitochondria and the cell membrane. And
Hmm.
if it exists beyond that limit, and we have a deficiency of antioxidants, and think in that way that I think oxidative stress will be more, and that the cell will be go a pathway of necrosis or maybe apoptosis.
Okay.
And I think it will be damaging to the body internally, having nothing going from the outside the like the body from the mouth or maybe any other way.
Okay, okay, Gurpreet. Okay, so I'm just coming to the first slide. Is this visible?
Yes, sir.
Yes, sir.
Okay, so actually, see, immunity is the state of resistance to the disease, infection, or any unwanted foreign invaders. It's not always that something from outside is attacking the body and the body is resisting. Any problematic change that is happening inside the body, if the body is resisting those changes also, that's also part of immune system. So immunity is the biological state where the body is ready to defend itself from anything that can cause problem. Okay, whether it's coming from outside or whether it's developing inside, whatever it is. And this immunity, the state of immunity, is orchestrated by the immune system that consists of a network of cells. There are a lot of cells which are involved in performing this immune action. There are tissues, there are organs that work together to protect the body. And therefore, immune system is a complex network of organs, cells, and proteins that defends the body against infection or non-infection injury. Any example of non-infection injury that body resists very regularly? Like as a normal thing, whenever we hear the term immune system or immunity, the first thing probably that comes to our mind is infection. But can anyone tell what is one non-infection condition that our body resists daily, almost every second, every fraction of second, the body is resisting that non-infection change? Anyone? Am I audible?
Yes, sir.
Okay, okay. So not
Yes,
taking
sir.
much time. It's cancer.
Okay.
Okay, every second, a lot of normal cells are being, or they try to become cancerous, but till the time the immune cells they are doing their job perfectly, they can recognize when a normal cell becomes cancerous, trying to become cancerous, they will kill that cell, right? So it is happening every second. A lot of normal cells are becoming cancerous, but because of the immune system, our body is safe. Okay, the biggest fundamental of immunity or immune system is the differentiation between self and non-self. If I may mention this in Hindi, it is distinguish to distinguish between apna and paraya. Okay, anything which the body considers, or the system of the body, the tissues and the organs consider self, the body or the immune system will not mount any action against that. And anything, whether it's coming from outside or it's developing inside, if the immune system consider it as non-self, or start considering it as non-self, it will mount an immune response against that. And this differentiation of self and non-self is the fundamental and is also responsible for autoimmune diseases. How many of you have heard of autoimmune diseases?
Yes, sir.
Right?
Yes, sir.
So, a very interesting
Yes, sir.
analogy. Okay. A very interesting analogy I always use for autoimmunity is that fire extinguisher itself got fire. Right? Like we keep fire extinguisher to switch off the fire, but if the fire extinguisher itself gets fire, that is an analogy for autoimmunity. That means the immune system of the body starts functioning against the body itself. Okay, and when we also hear about immune system, uh, there are two things again that come to our mind very frankly, very commonly. That is antigen and pathogen. Uh, can anyone tell me the difference between antigen and pathogen? Anything that comes to mind.
Sir,
See,
antigen is usually which for against antibodies formed in our cell, and pathogen is anything that is causing disease or infection in our body.
okay. So people who are suffering from asthma, if they regularly go out into a dusty environment, they get disease in their body, right? Disease in form of various symptoms. But in this case, dust is causing those symptoms. So what kind of classification will can we put the dust into, antigen or pathogen?
Sir, antigen.
Pathogen.
Antigen. Now, based on this example, can anyone tell me again the difference between antigen and pathogen? See, I always tell in my class also, fundamental science is completely based on the terms. Using the correct term is extremely important in fundamental science. Okay, and not only because or just because of use, it will sharpen our fundamental knowledge. Okay, so anyone, whatever comes to mind, any difference between antigen and pathogen?
Sir, pathogen are pathogen can be a living organism, a bacteria or a virus, whereas
Mhm.
a antigen can be any biological molecule or any non-living particle.
Exactly, exactly. So all the pathogens are antigen, but all the antigen may not be pathogen. Simple. Any living entity, though we cannot place virus into a living or non-living, it's a particle. Virus is also always called a particle. Any pathogenic microorganism, whether it's fungi or bacteria or virus, if we take virus into that, they can be called as pathogen, even intracellular parasites or extracellular parasites. But anything, but antigen can be a non-living entity, like the example of dust that I just mentioned. You might be knowing that a lot of people are allergic to peanuts or onion or egg white, so they are also causing a lot of symptoms in the body. They can be placed into antigen, may or may not be, but they can never be placed into the category of pathogen, right? So I hope all of you are clear about antigen and pathogen.
Yes, sir.
Okay, great. So when we classify immune system, the three major classification that we come across is innate and adaptive, active and passive, cellular and humoral. But out of these three, the last two, active, passive, and cellular humoral, are based on innate and adaptive. Innate immune, the components of innate immune system are something which are present since birth, and they don't differentiate between whether it is coming from outside or inside, or it is pathogen or antigen. If they are mounting a response, they will mount the response, a similar response for everything that they come across. They are present since birth, first thing, and they don't differentiate between the antigen. Okay, some major components of innate immune system are macrophage, dendritic cells, natural killer cells, eosinophils, basophils, monocytes, mast cells, skin, mucus, and complement system. So if like you might have heard that the skin is the first line of defense for anything coming from outside, apart from the vestibules. If a skin can resist entry, I'm talking about the intact skin, not of a wounded skin. So if an intact skin can resist the entry of bacteria, it will, in a similar, very similar fashion, resist the entry of fungus also. Okay, so innate immune system components, they don't differentiate between anything. Uh, the B cells and T cells specifically are the part of adaptive immune system. There's another category of NK cell and T cell, which is in between NK cell and T cell, called NK T cells. They are also a part of innate and adaptive both, but B cell and T cells are very specifically a part of adaptive immune system. And any part of adaptive immune system, whether the cellular or the proteinaceous part, like antibody is a part of adaptive immune system that helps the innate immune system a lot. Okay, so any part of the adaptive immune system is highly dependent on the components of innate immune system. Okay, when we talk of innate and adaptive is, you may say, this classification is based on whether the components are present since birth and they are able to distinguish or not. But when we talk of active and passive, it is based on the action. So the first component of active immunity is antibody. If you can see the slide, I have written immunoglobulins in bracket. Okay, now can anybody tell me what is the difference between antibody and immunoglobulin? See, never hesitate to speak. The maximum what can happen is that your answer will be wrong, nothing more. है ना? And if you can believe me, science is all about failure. There is very less success in science, and that's the beauty of science. That teaches us a lot. So don't worry about giving anything that comes to your mind. Anyone? Difference between antibody and immunoglobulin? Okay, I'll come to this later when we'll be discussing about B cells and antibody. But just for now, remember, when immunoglobulins come into circulation, they are called antibodies. But there are immunoglobulins which are a part of the cell surface of B cells, immature B cells. In that condition, we'll call those proteins as immunoglobulins, not as antibodies. If we go to technical terms, okay. Memory cells. You might have heard of B memory cells and T memory cells. Most of the memory cells in our body is that of B cell. They are a part of active immunity. But you might have heard that in case someone is bitten by a rabid dog, we may not be knowing whether a dog is a rabid or not, but in case there is a dog bite, the person is given vaccine and antibodies. These are prepared antibodies. Okay, you might have taken antitoxoid in case of tetanus. So these are pre-prepared toxoids. Okay, so when we administer these antibodies or toxoids into a person, so that the body of the person can fight the infection or anything that is causing the problem, this activity comes under passive immunity. That means these are pre-prepared things which are given into the body. The third classification of immune system is cellular and humoral. All the cells you have been studying since your school days, white blood cells are responsible for immunity. So any cell that comes under the classification of white blood cells or leukocytes constitute the cellular immunity, while any protein or non-protein component that are part of immunity comes under humoral immunity. And this classification has come from the term humor. Humor means fluid. Now, anything which is secreted from the immune cells, whether it is antibody or cytokines or chemokines, they come into circulation. They are not located into a particular they don't sit idle at a particular tissue or organ. But a lot of cells sit on different cellular immune organs, like lymph nodes and spleen. Like B cells are primarily found in spleen, while B cell and T cells both together are found in lymph nodes. So they are the site for these cells. But for anything which is secreted by the cells will be in circulation, whether in blood circulation or whether in lymphatic circulation. And because of the circulation, because of the fluidic nature of the circulation, this component of immune system is called humoral immune system. Okay, so this is what we were discussing. So in innate immunity, we have epithelial barrier, which is skin. We have non-differentiating cells. That means not in terms of cellular differentiation. They don't differentiate between whatever comes in the body. They are macrophage, neutrophils, dendritic cells, natural killer cells, antibodies which are formed, and complement system. Though antibodies formed from B cell, but once in circulation, they help the innate immune system or the components of innate immune system to function properly. And that's why, not very strictly, but you can place antibodies in the adaptive or the innate immunity arm. In the innate immunity, in this particular picture, you can see it's written natural antibodies. That means, okay, which antibody can cross placenta? Anyone?
Sir, I guess IgG.
IgG. Okay, and there are 4 isotypes of IgG. Which IgG can cross placenta specifically? Like there is IgG 1, 2, 3, and 4. Anyone? Which one can cross placenta?
Sir, I'm not sure. Maybe type 2.
Yeah, exactly. It's type 2. Okay. And in the adaptive immune arm, we have B cells, T cells. You already know there are 2 types. Basically, we read about 2 types. There are not only 2 types of T cell, but generally we read and hear about 2 types of T cell, T helper and cytotoxic. Okay. So innate immunity is present by birth. It is non-specific. It is the first line of defense, acts on antigens in the same manner. While adaptive immunity shows specific action towards antigens or pathogens, such as dust or bacteria. So if a person is exposed to dust, IgE will be released from mast cells or basophils. But if a person encounters pathogen, the first immune cell that resists the pathogen are the resident phagocytes, which primarily consist of dendritic cells and macrophages. And that's why it is mentioned, adaptive immune systems show a specific action. If it is antigen, there will be a different set of cells which will be activated. If it is a pathogen, there will be a different set of cells which will be activated. And the adaptive immune system, a lot depends on antigen presentations and antigen presenting cells. We'll discuss about these cells later on. Okay, now come to macrophage firstly, under the category of immune cells. So macrophages are very specialized cells which, whenever we hear about macrophages, the first thing that comes to our mind is phagocytosis. But macrophage is not only responsible for phagocytosis. There are 4 very important functions, almost equally important to phagocytosis, that is done by macrophage. So there are specialized cells involved in detection, phagocytosis, and destruction of bacteria and other harmful organisms. They originate from the blood monocytes. So the primary source of most of the macrophages in our body is the monocytes. Monocytes, they are the kind of immune cells which are normally in the blood circulation, but when they receive a call from a tissue that encounters a pathogen, on receiving the call, monocyte goes to the tissue and becomes macrophage. So there are a lot of morphological and protein-based changes, cellular changes in the monocyte, that they become macrophage once they reach the tissue that have called those monocytes to resist the infection. Macrophages migrate to and circulate within almost every tissue, patrolling for pathogens or eliminating dead cells. You might have seen police vans in your colony or in the city doing patrolling very regularly. Macrophages do such kind of patrolling throughout the body very frequently. Macrophages are essential. So, though macrophages are not in circulation, when I say macrophages patrol, that means, see, monocyte, when monocytes enter the tissue, they become macrophage. Now, one of the primary role of macrophages in tissue is to collect the pathogenic sample, take the sample to the lymph node, and then activate the T cell and B cell. Because in the tissues, suppose there is a infection in skin. Now you won't find B cell and T cell in the skin mostly, right? So one of the role of macrophages or dendritic cells in tissues is to collect the pathogenic sample. I'm telling sample, not all the pathogenic cells. They collect the sample, take the sample to the lymph node, and lymph node are the major playground of the immune system. Okay, so they do these kind of patrolling. Macrophages are essential for maintenance and defense of the host tissues, doing so by sensing and engulfing particulate matter and, when necessary, initiating a pro-inflammatory response. Now, see, whenever we hear the term inflammation, uh, if I'm not wrong, most of the time we get a negative sense of inflammation. But believe me, a range of inflammation is highly important and very important part of normal immune system. Okay. During organogenesis, macrophages derived from the embryonic yolk sac and fetal liver precursors are seeded throughout the tissues, persisting in an adult as resident self-maintaining population, which turn over locally under steady state condition. Okay. I recall one very common question, which is the first cell that that gets activated against an infection? Anyone? Which is the first immune cell that gets activated in response to an infection?
Is it leukocytes?
Every cell which we are discussing is leukocyte. Okay. How many of you, because I cannot see the raised hand, you can say yes or no. How many of you have read or you may recall that neutrophils are the first cell that respond to infection? You can just say yes or no. I am not able to see
Yes.
the hands.
Yes, sir.
Okay. One answer. Anyone else? You can mention yes or no. I cannot see the hands. Okay. So I just got one answer verbally. Uh, you can recall that neutrophils are the first cell that respond to infection. Okay. Now, as I said earlier, terms and sentences are very important in fundamental sciences. Okay. So when I say the first cell that respond to an infection, believe me, they are the resident phagocytes. Okay. Resident phagocytes are mostly macrophages and dendritic cells, and they are the part of the tissue. They are resident, they are the local resident of the tissue. As soon as the tissue is invaded by the pathogen, these resident phagocytes, macrophage and dendritic cell, they take the pathogenic sample cells and move to the lymph node. In the lymph node, they destroy the pathogen, present the component or the fragment of the pathogens over their surface through MSC2. And because the fragment of the pathogens are exposed through MSC2, it will call and bind to the T cell, inactive T cell. And because it is MSC2, the T's inactive T cell will become T helper cell. And one of the function of T helper cell is to help B cell getting activated. Once the B cells are activated, if they when they become plasma B cell, they will start producing antibodies. And this is how we get antibody against different pathogens. So the first cell that actually respond to the pathogens entering a tissue space are the resident phagocytes. They collect the sample, move to the lymph node, and activate the adaptive immune arm. Now, when there is a when there is a pathogenic infection in the tissue, the epithelial cells and these resident phagocytes, they also release certain chemokines and cytokines and uh, send them into the circulation. These chemokine and cytokine are the call to other immune cells that there is a problem in this particular tissue, come and defend that. Now, based on the call from these chemokines and cytokines, the first cell that enters the tissue space because of the infection are the neutrophils. But the first cells that respond to the infection or the pathogen are the resident macrophages or the resident phagocytes. Is this part clear or any doubt?
Yes, sir. Clear.
Okay, great.
Yes, sir.
So unless until there are resident phagocytes, the neutrophils won't reach the site, right? Okay. Now, the last paragraph which I was discussing. So you might be knowing that during the fetal stage, the development of blood cell or hematopoiesis is restricted to embryonic sac and liver. From the seventh month, the the part starts going to bone marrow, but mostly till the seventh month the hematopoiesis is the result of action of embryonic yolk sac and liver. Now in this duration only, when the hematopoiesis is happening in yolk sac and liver, these resident phagocytes are seeded into different tissues, so that when the child is born, the tissues already have the resident phagocytes to tackle the infection. After the birth, so the last sentence I'm reading out. After the birth, the bone marrow derived blood monocytes replenish resident macrophage population with high turnover. Larger numbers are recruited following injury, infection, and sterile inflammation, and give rise to infiltrating activated tissue macrophages. Okay. Now, anything that is born is bound to die. Similarly, any cell or any protein which is there in the body will definitely have a fixed lifespan, right? So when these macrophages or dendritic cells are seeded into the tissue during the fetal development as resident phagocytes, they do have their own lifespan. They cannot be present in the body till the last breath of the person, right? And that's why to replenish the population of the resident phagocytes, monocytes, based on a lot of inflammatory reactions, they move from the blood to the tissue space to replenish, to top up the amount of macrophages present in the tissue. Is this clear?
Yes, sir.
Okay, great. Yes, sir. Okay. So monocytes leave the circulation squeezing through the blood vessel wall in a specialized process called diapedesis. So whether it's monocyte or neutrophil or any cell, any immune cell that on call from the chemokine or cytokine, when they're trying to move to the tissue space. See, monocyte or neutrophil, whether it is a monocyte or neutrophil, they are present in a normal circumstance, they are present in the blood circulation. But when they get a call from the circulation, they need to move to the space from where they have got a call. Now, between the blood circulation and the tissue space, there's a barrier which is called the blood vessel, right? And there are three layers of blood vessel. Can anyone tell me the three layers of blood vessel that we have been knowing till now?
Sir, one is tunica intima, tunica media, and tunica exterior.
Externa. Very good. Very good. Yeah. So tunica externa and tunica media are made up of smooth muscle cells, while tunica interna is the system that we very commonly know as endothelial layer. Okay. That means when a monocyte or neutrophil gets a call to move to a tissue space to tackle the infection, they must cross these three layers to move to the tissue. And this movement of WBC or leukocytes from the circulatory system to the tissue space is called diapedesis. And this is done to give rise to heterogeneous tissue macrophages in case the movement is that of the monocyte, or they can remain within the blood vessel to help maintain the endothelium. One of the major roles of circulating monocyte is to maintain the health of endothelial layer. Macrophages are not cells exclusive to the immune system. They also play a central function in many other aspects of embryonic development, homeostasis, and wound repair. Homeostasis means? Anyone? What do we mean by homeostasis?
So we do have an external environment and internal environment. So when the internal environment is maintained with the changes, physiological changes event happen to the external environment, that is known as a homeostasis.
Great, good. So resident macrophages become adapted to perform particular function, and in different organs, we have just named them differently. Like in brain, the macrophages are called microglia. In the lungs, macrophages are called alveolar macrophages. In the liver, they are called Kupffer cells. Okay, these are different kind of macrophages in different location. Alveolar macrophages in lung, Kupffer in liver, microglia in the sinus, splenic macrophages in spleen, interstitial macrophages in the gut, osteoclast in the bone. What is the role of osteoclast? Can anyone tell me? Though it's written. So it maintains the overproduction of bone cells. Okay. We have sinus histiocytes in lymph nodes. We have Langerhans cells in the skin. Even though the dendritic cell and macrophage both in the skin are called Langerhans cells, and we have Hofbauer cells in the placenta. These are the macrophages present in different tissue locations that help fight infection in that particular tissue. Okay, so when we move on to the four major functions of macrophages that are known till now, I may be repeating this term known till now, because one of the most beautiful aspect of science is that nothing is permanent in science. What the theory which we know today may be discarded tomorrow. Okay, and science is ever developing. So whatever thing we are studying today are just the information that we have got till today. There are no absolute truth in science. Okay, so the four major function of macrophages that we know till today are antigen presentation. They present the antigen or pathogen to the respective T cell so that they can mount the immune action. They help in phagocytosis, they help in immunomodulation, and they help in homeostasis to maintain the normal state of the body. So first, let's discuss about phagocytosis. So you all might have heard about phagocytosis. It is a kind of eating by certain cells, and when we call eating, that means the thing which is eaten up is something solid. Okay, another component of I may not call it eating, but drinking by the cells is pinocytosis. So any liquid component which is present outside the cell, if it is taken inside, that is pinocytosis. Okay, so macrophages ingest materials to eliminate the waste and debris and to kill invading pathogen. The microbicidal mechanism of phagocyte are largely confined to intracellular vesicles called lysosomes and phagolysosome. Now, because the action is phagocytosis, and because of the action, the cells are called phagocytes. As soon as a phagocytes, I'm not only talking about macrophages, I'm talking about all the phagocytes, any WBC that shows the action of phagocytosis. Whenever a phagocyte engulf a material present outside, the vacuole or the balloon-like structure that is formed inside that carries the pathogen, suppose in this case, is called phagosome. Now, once the phagosome is completely formed, it fuses with the lysosome inside the cell. And you all might know, we have been studying since school, lysosomes, they are the cellular structure that contains a lot of hydrolytic enzymes. And because the enzymes are so crucial in breaking down almost all the macromolecules, lysosomes are also called suicidal bags. Because if they burst out, the cell will be dead. So the phagosome fuses with lysosome, and the new structure formed is called phagolysosome. Now, this new phagolysosome is the structure, it's a temporary structure. But because of the fusion of lysosome and phagosome, the phagolysosome contains the enzymes as well as the pathogens. Now, because the pathogen is in the vicinity of the hydrolytic enzyme in the same vesicle, these enzymes destroy the pathogen. This is the major mechanism of phagocytosis that we know till now. Macrophages also express receptors that activate the cells to produce cytokines and microbicidal substances and receptors that stimulate the migration or chemotaxis of the cells to the site of infection. Macrophages have also got receptors over their surface for opsonins. So how many of you have heard of the process called opsonization? Can anyone tell me what is opsonization? Hello? Am I audible?
Yes, sir.
Okay.
I think
Anything that comes to mind?
opsonization is like kind of intensification of phagocytosis. I don't clearly remember. Like it
Okay.
actually induces phagocytosis.
Okay. Not an issue. Um, how many of you have lived in a colony structure, not in the high-rise apartments? Maybe in your childhood or even today. You can respond by saying yes or no. Anyone
Yes,
living in
sir.
a colony structure and not in high-rise apartments?
Yes, sir.
Okay.
Yes, sir.
Okay. Can you recall that when the municipal corporation needs to widen the road of that colony, they would inspect uh, about which houses have their boundary walls extending more than the allotted space, and they would just uh, put some uh, uh, put some indication over the boundary wall so that when the bulldozer comes, they will only destroy those boundary walls and will leave the rest, which are as per the rules. Mostly, the people, they put a cross mark over the boundary wall that need to be demolished. How many of you have seen this or can recall this?
Yes, sir.
Okay, so suppose there are 20 houses in a colony and 10 of the houses have their boundary walls extended on the road. That is against the rule. That means when they wish to broaden the road, the municipal corporation needs to demolish that portion of the boundary wall so that the road can be extended in size or width. And for that purpose, they put a indication on the boundary wall, mostly in the form of a big black cross mark. They put a X over the boundary wall so that when they send the bulldozer, they will only destroy or demolish the boundary wall that have this big black cross mark over them. That means they have put an indication that only these boundary wall need to be destroyed. The rest of the boundary wall of the houses can be exempted. Opsonization is a similar kind of putting a recognition mark over the pathogens. Okay, now this recognition is done by antibodies. So the antibodies that are finally released by the activated B cells, they come into circulation and find the pathogens that have very similar epitopes. The epitope for which the antibodies have been released, they try to find the pathogens with a very similar epitope or maybe the same epitope. Once these antibodies find the same or similar epitope, they go and bind to the epitope. You might recall that antibodies bind to the antigens, and the site of binding is exactly between the paratope of the antibody and the epitope of the antigen, right?
Yes, sir.
Okay, so this attachment of antibody over the particular pathogen is called opsonization, and these antibodies are called opsonins, right? So if you can recall in the previous slides, I have mentioned that the resident phagocytes, they collect the pathogenic sample, move to the lymph node, destroy the pathogen, present the fragment of the pathogen over their surface through MSC2. This presentation, after the attachment of T cells, activate the T helper cell, and the T helper cells activate B cells to release antibodies against the fragments which are exposed over the surface of the macrophages. Now, these antibodies which are released into the circulation, they will try to find the fragments for which they have been activated or they have been released. Once they find the fragment over the live pathogen, they go and bind to the surface of the pathogen. Now, this complex becomes the antigen-antibody complex. And one of the major role of macrophage is to find out this antigen-antibody complex and destroy the whole complex. Right? Why is it so? Because see, macrophages, as it's written over there, they have particular receptors for these antibodies, which are called opsonins. Now, macrophages, though they can perform phagocytosis even in the absence of the antibodies, but they can only pick some number of pathogenic cells. But once they find a lot of these antigen antibody complex antibodies over the surface of the pathogens, this initiate high-end phagocytosis from the macrophage. That means if macrophage is present in a population of 100 bacteria, it may just phagocytose 10 of the bacteria while the other 90 bacterial cells remain as it is. But if there are antibodies bound to the surface of all the bacteria, it will be very easy for the macrophage to initiate a high-end phagocytic response, and it will eliminate most of the bacterial cell. And that's why opsonization is the first thing, or is the most important thing a macrophage is looking for. So the macrophage, surface of the macrophage has receptors for opsonins that promote the phagocytosis of microbes coated with various proteins such as antibodies and complement proteins. FC receptors and complement receptors of macrophages contribute to the opsonin-mediated process. Okay. How many of you can recall the structure of antibody? Though we will be discussing in next further classes, but you might recall that so it's a Y-shaped molecule, right? And the two fangs of the antibodies are the portion that will bind to the epitope of the antigen, while the single lower portion of the antibody is called the Fc receptor. That means it is the Fc portion that binds to the Fc receptor present on the surface of cells such as macrophages. Okay, so once let me try to. So suppose this is a bacterial cell, and this is the antibody that is bound to the bacterial cell. Okay, now this is the Fc portion of the antibody. This is the FAB. Okay, you might have heard of FAB, that is fragment antigen binding, and this is the Fc portion. The single downward portion is Fc portion. Now this is the antigen antibody complex. This is the antigen and this is the antibody. Now this whole complex, when a macrophage through the Fc receptor sees this complex, antigen antibody complex, this is the macrophage, it will engulf the whole antigen antibody complex inside. Okay, so this is how a macrophage initiates phagocytosis. Any doubt in this?
No, sir.
No, sir.
So this attachment of antibody over the surface of its respective antigen is called opsonization. Okay, so once the macrophage will phagocytose the pathogen, only then it can present the fragment of pathogen over the surface through MHC2. There are two kinds of receptors present on the surface of macrophage. One is in terms of antigen presentation, there are MHC1 and MHC2. If you can recall, all the nucleated cells have MHC1, which can present the protein fragments on the surface, while antigen presenting cells, they are the specialized cells that can express MHC2 on their surface. Now, any phagocyte is a APC, antigen presenting cell, whether it's a macrophage or a dendritic cell or a neutrophil or a B cell. If you can recall, B cells are very potent antigen presenting cell. Okay, so one aspect of activation of B cell is through T helper cell, where a T helper cell is activated by the macrophages or dendritic cell, but B cells can activate themselves by directly interacting with the pathogens or antigens. Okay, so once the macrophages have phagocytosed the pathogen, they take the pathogen to the lymph node, they destroy the pathogen in the phagolysosome, and once the pathogen is destroyed and broken down into different fragments, these fragments are randomly, I'm saying randomly because multiple fragments can be exposed on the surface of a single macrophage or dendritic cell. The fragments are actually exposed based on their size, which we will be discussing when we will be discussing about antigen presentation. It's a separate topic. So all these fragments are randomly exposed on the surface of the macrophages through MHC2, major histocompatibility 2. And once the antigen fragments are presented, the adaptive part of the immune system that consists of T cell and B cells are activated. And that's why in the very first slide we have discussed that adaptive immune response is only or largely dependent on the innate immune response. Is this slide clear?
Yes.
Okay, great. The third very important aspect of macrophages is immunomodulation. They don't only function for phagocytosis or antigen presentation. They are very important cell for immunomodulation. So, like activated macrophages release cytokines such as interleukin 1, 6, TNF alpha, interferon, alpha beta both, interleukin 10, 12, interleukin 18 that participate in the regulation of immune or inflammatory responses, such as interleukin 12 is a heterodimeric cytokine produced primarily by antigen presenting cells, whether it's a macrophage or dendritic cell or a neutrophil or a B cell, that has important activities in the regulation of various aspects of immune response. Interleukin 12 stimulates the proliferation of activated T and natural killer cells. It enhances the natural killer cells activity and lytic activity of cytotoxic T lymphocytes. It induces the interferon gamma production by the T cell and NK cell. Okay, why are interferon gamma very important? Like to tackle what inside the cell? Anyone? Why do we know interferons for?
Sir, virus infection.
Exactly, exactly. And see, when a cell is infected by virus, the two primary cell that come in response to the virus infected cells are the NK cells and the T cells, right? Now these will get activated and destroy the viral infected cells also, and produce interferon gamma only when they are activated by interleukin 12. Okay, this is just one example where I am just showing that these interleukins and cytokines, they are very important in controlling the function of all the WBCs. And because these cytokines or chemokines are released by these APCs, not only macrophages, these APCs or phagocytes are very important cells that function in immunomodulation. Okay, because we are talking about macrophages, we are also talking about homeostasis. That means they maintain the body by destroying anything present inside that can cause problem for the body. Okay, one of the major role in maintaining the homeostasis is that macrophages can destroy the dead cells. Like they are the primary cells, more important than dendritic cells, that destroy the dead cells. Okay, because see, when a cell is live, the immune system will recognize the cell as a normal cell or any normal entity. So when it is a live cell, the immune system will consider it as self. If you can recall the earlier slide, the first slide, we have discussed the most fundamental thing that the whole immune system is based on is the distinction between self and non-self. So when a cell is live, most probably the immune system will consider a live cell as self, and there won't be any autoimmune response against that. But when a cell is dead, the immune system is not going to recognize it as a self. It will always recognize it as a something non-self, even though it is a part inside the body. But because it is dead, the immune system is not trained to recognize dead cells as self. They are trained to recognize dead cells as non-self. So as soon as a cell is dead, there are a lot of morphological changes that happens. One of the major morphological changes that is sensed by the macrophages is presence of certain pattern which is called DAMP. It is also written in the last paragraph. That is damage associated molecular pattern. Any pattern which is present on the damaged or dead cell but not on the live cell. One very basic example is the exposure of phosphatidylserine or PS. Can anyone tell me what is phosphatidylserine? Or anyone of you heard of phosphatidylserine?
Yes, sir. It is present in the membrane bilayer as a
Very good.
component, and
Exactly.
usually
Exactly. It
it's
is one of the.
the location.
Yeah, please go ahead.
Sir, the location of this phosphatidylserine in inner leaflet or outer leaflet actually determines if the cell is healthy or not.
Very good. Very good. So it is one of the major lipids present in our cell membrane, right? In a normal cell, in a live cell, PS is present inside, like on the inner side of the cell membrane. Now, when a cell is dead, it is exposed outside. Now, the immune cells are not trained to recognize something called phosphatidylserine on the surface because they have not seen anything like that when they were getting activated. Now, as soon as, suppose we are talking about macrophage, as soon as a macrophage sees phosphatidylserine on the surface, it very promptly respond that it is a dead cell or a damaged cell. It's not a normal cell, and therefore presence and recognition of phosphatidylserine is one of the damage associated molecular pattern. As soon as macrophage recognize any such kind of damp, it will engulf the damaged cell or the dead cell, so that there is no or hardly any inflammation caused because of the accumulation of dead cells. Like every second, because see, cell death is a part of cell turnover. It is a very normal thing for the body. But if the immune system not working properly, particularly macrophages, the dead cell will keep on accumulating in the body and will result in a very toxic inflammation inside the body. And that's why, until and unless our macrophages are working properly, our body will not have any issue with the dead cells, because the macrophage will detect the damps and will clear off the dead cells. Is this understood?
Yes, sir.
Okay. Now, this eating of dead cells by the macrophages is called efferocytosis. When a macrophage eats something from outside, mostly in case of pathogen, we term that process as phagocytosis. But because this removal of dead cell is very unique, and the dead cells are not live pathogens, we have just given a different name to this process called efferocytosis. Okay, similar to damp, because see, macrophages are finding something very unique on the surface, based on which they have decided to remove the dead cells. Right? Suppose it's phosphatidylserine. Macrophages, we have discussed, and you all know, eats up the pathogen present outside the macrophage in a process called phagocytosis. So how do they do that? The one thing that we discussed in the last slide was opsonization. So when a macrophage find a antibody stuck to the surface of a pathogen, it will destroy the complete antigen antibody complex. But what if there is no antibody present over the surface of a pathogen, suppose bacteria? Now there are a specific signature present on the surface of bacteria similar to damp. There are specific patterns present on the surface of, say, bacteria. I'm just taking the example of bacteria, which indicates the macrophages that I am not a normal thing for your body where you're residing. You can eat me up. Such patterns, which are recognized over the surface of pathogens by the phagocytic cells, are called PAMP, pathogen associated molecular pattern, PAMP. One very common example of such a pattern is lipopolysaccharide layer present over the gram-negative bacteria, LPS layer. I hope every one of you recall LPS layer over the gram-negative bacteria.
Yes, sir.
So
Yes, sir.
LPS is a very prominent PAMP, pathogen associated molecular pattern. Any molecular pattern which is unique to a pathogen. Like LPS will never be found on the surface of a gram-positive bacteria or any other pathogen such as fungus. Will always be found over the surface of gram-negative bacteria. So there will be a different kind of response for presence of LPS. For a gram-positive bacteria, the NAG and NAM, which are the component of the cell wall, are the PAMP, pathogen-associated molecular pattern. Presence of ergosterol or presence of chitin over the cell wall of fungus is PAMP for the fungus. So we have just discussed, for a dead cell, there are unique features which are now present on the surface of dead cells, which are recognized by the macrophages, and based on these damage associated molecular patterns, the macrophages clear off the dead cells to maintain the homeostasis. In case of a pathogen, these macrophages, for example, because we are discussing about macrophages, or it is true for any of the phagocytes, they detect a specific kind of molecular pattern over the surface of pathogens and they clear off the pathogens. Such molecular pattern present on the surface of pathogens are called pathogen associated molecular pattern, or PAM. Now these DAMP or PAMP, because they are a physical entity, they need to bind to a receptor over the surface of phagocytes. For example, macrophage for this particular example. That means the components such as phosphatidylserine, it will act as a ligand for its particular receptor over the surface of macrophage. LPS will have its own receptor on the surface of macrophages. Now, these receptors that recognize and bind to DAMP or PAMP are called pattern recognition receptor, or PRR. So these are the receptors that recognizes a particular pattern over the cells which need to be cleared off, whether it's a dead cell or a pathogen. So there will always be a joining, a physical joining between PRR and DAMP or PAMP. And based on this ligand receptor interaction, the macrophage will clear it off. Is this clear?
Yes, sir.
Everyone? I'm hearing only one voice.
Yes, sir.
Okay, great. Thank you. Please be interactive. That will help me in knowing how far I can go, how in depth I should go. And like interactive class is always better than a one-way monotonous class. Okay. Okay. So these are the four functions of macrophages. Phagocytosis, they clear out the pathogens. They present the fragment of pathogen over the surface to activate the adaptive immune response. They perform a lot of immunomodulation so that the immune cells can function normally, and macrophages maintain the homeostasis of the body. Any doubt in these four functions? Any doubt?
No, sir.
Okay, great.
No, sir.
Great.
No, sir.
Great. Okay. Macrophages that encourage inflammation are called M1 macrophages, whereas those that decrease the inflammation and encourage tissue repair are called M2 macrophages. There is a bit of debate where there is such a classification of macrophage or not, but normally we do study about these two classes of macrophages. So macrophages that encourage the inflammation, that means they will release the pro-inflammatory cytokines. As we just discussed a few moments earlier, that a bit of inflammation is very important for the immune system to for normal function, because a lot of pro-inflammatory cytokines, they can directly tackle a lot of dead cells or pathogens. So macrophages that releases pro-inflammatory cytokines are M1 macrophages, while those that releases anti-inflammatory cytokines and encourages tissue repair are M2 macrophages. This difference is reflected in their metabolism, such as M1 macrophages have a unique ability to metabolize arginine to the killer molecule nitric oxide, whereas M2 macrophages have the unique ability to metabolize arginine to convert it into a repair molecule called ornithine. That means you can see when arginine is metabolized in the macrophages into nitric oxide is a very potent pro-inflammatory molecule that destroys the pathogen directly. Okay, so when arginine is metabolized to nitric oxide in the macrophage is in the M1 state because it is encouraging the inflammation and maintaining the immune system. While the same arginine, if it is metabolized to a tissue repair molecule called ornithine, we can say the macrophage is in M2 state because it is in the repair mode. The names M1 and M2 were chosen because M1 and M2 macrophages promote Th1 and Th2 responses respectively. If you may recall, we have been studying the two common types of T cells as T helper and T cytotoxic. Among the T helper cell, we study about Th1 and Th2. So Th1, T helper cell 1, Th1 cells promote inflammation, while Th2 cells discourages the inflammation or maintains high inflammation or regulates the high inflammation, and they also help in tissue repair. And because different state of macrophages help promote the activation of Th1 or Th2, these states are respectively called M1 and M2. Interferon gamma can differentiate macrophages into M1 macrophage that promote the inflammation, but IL-4, interleukin 4, produced by Th2, can convert the same macrophage into M2 type macrophage that inhibit the inflammation. That means when the level of inflammation goes beyond the threshold level or goes beyond the level that is required for the normal functioning of immune system, if the level gets increased, the macrophage turns to M2 type and inhibit the excess inflammation. The role of M1 macrophages is to secrete pro-inflammatory cytokines and chemokines, present antigens over the surface, and thus participate in the positive immune response and function as immune monitor. The main pro-inflammatory cytokine that is produced by M1 macrophage is IL-6, IL-12, and TNF-alpha. M2 macrophages mainly secrete arginase 1, interleukin 10, and TGF-beta and other anti-inflammatory cytokines which have the function of reducing the inflammation and contributing to tumor growth and immunosuppressive function. This is very important. Though whenever we hear the term inflammation, there is something negative comes to our mind. As a general brain processing, we think inflammation something as very negative. But as we have discussed, a threshold level of inflammation is extremely important for normal immune function. And therefore, because M2 macrophages are anti-inflammatory, you will find a lot of macrophages in most of the tumors in their M2 state. Because, see, when a normal cell start becoming tumorous or cancerous, not only macrophage or not only T cell that comes to rescue. There are a lot of immune cells that comes to destroy those cells which have become cancerous. One of the major action through which these immune cells destroy the cancer cell is by releasing cytokines and pro-inflammatory chemicals. And therefore, a normal cell that has become tumor cell will not want itself to be killed by any mode. And that is why it secrete, like it is an established fact till now, tumor cells or cancer cells, they secrete certain molecules that converts the M1 macrophage into M2 macrophage. Now, because the macrophages around the tumor cells are in M2 state, they are anti-inflammatory. That means they will not destroy the tumor cells. So even though M2 macrophages are very important in tissue repair, suppose in case there is a open wound and the wound needs to be repaired, it's M2 macrophages that will help in wound repair. But the same M2 macrophage, because it is anti-inflammatory, will also contribute in the growth of the tumor, because tumor cells have converted the M1 macrophage into M2 macrophage. Is this classification clear? Any doubt?
Yes.
Any doubt up till now? Please speak.
Sir, this M1 and M2 macrophages we are talking about, the single macrophage can shift into two state,
Yes,
or they are
yes,
very.
yes. Is that just two state? They are not different macrophages. The two state of the macrophage. If a macrophage is producing pro-inflammatory cytokines, we will call the macrophage as M1. If it is producing anti-inflammatory cytokines, we will call macrophage as M2. If the macrophage is converting arginine into nitric oxide, we will call macrophage as M1. If it is converting the same arginine into ornithine, we will call macrophage as M2. It's just the change of the state.
Sir, I have one more question.
Yeah.
How is the nitric oxide? I can understand it is like highly reactive, but how ornithine is playing a role in tissue repair? Can you just briefly explain?
Yes. Okay. See, you might have heard of the term wound. It's a very common term.
Yes,
Right. There are two types of like when we classify wound, there are different classification of the same thing in biology. One of the classification of wound is open wound and closed wound. All the open wounds are extremely susceptible to infection, right? And
yes,
any open wound, if it is infected beyond repair, we call those wounds as chronic wound. That means it cannot be healed naturally because of the presence of a lot of infection, and therefore it requires medical help. Okay. Now, because of the infection and because of the severe damage to the tissue, the automatic repair is beyond control. Like normally, when you have a small scratch over the skin, it repairs itself,
yes,
right? But if the wound is such severe that the wound is highly susceptible to infection, and not only infection, the severity or the damage to the tissues is such that the tissue cannot repair itself. At that point, the see, even if there is no infection, any tissue which is damaged, or if there is any kind of open wound, the wound will naturally try to get itself repaired, as we just discussed about a small scratch. So when a open wound is repairing itself, one of the molecules that helps in that repair is ornithine. Like what happens
okay,
when a wound is repaired? There are four phases of wound healing. In the first phase, so if there's a damage to the capillaries, there will be bleeding in an open wound. But you may find that for small wounds, the bleeding stops naturally,
yes,
right? And therefore, the first phase of any wound healing, open wound healing, is hemostasis, not homeostasis, it's hemostasis. The second phase is inflammation. That means because once before the bleeding stopped, the wound was open, there are high chances of pathogens going inside,
yes,
right? And that's why the second phase of wound healing is inflammation. That means the immune cells they will mount a level of inflammation that will help killing the pathogens. And there, the role of pro-inflammatory macrophages come. That is M1. Now, once the the pathogens that have entered the space, now they are cleared off. See, unless until the pathogens are completely removed, the tissue cannot be repaired. Because pathogens, see, it's particularly the bacteria that enters a wound space, open wound. These bacteria, they release chemicals that does not allow the wound to repair itself, right? So, and therefore, the second stage of wound healing is inflammation. The macrophages and other cells in that space, they will mount inflammation so that the pathogens can be killed. Now, once the pathogens are killed, the third stage is repair. And in that stage, when the tissue the wound sees that the pathogens are killed, now it's the time to repair myself. They will require a lot of ornithine for cellular repair or tissue repair. And now, because the pathogens are killed, a level of inflammation is not required now, and therefore the wound will convert M1 macrophage into M2 macrophage. That means they will indicate the macrophage that now your role as pro-inflammatory macrophage is over. Please come down to anti-inflammatory stage and metabolize arginine to ornithine so that I can repair myself. And the fourth stage sometimes is a scarring. You might have seen that once the wound is completely healed, there is a presence of scar. Right? Is
Yes,
this
sir.
clear?
Yes, sir.
Yes. So in second stage of wound healing, where there is inflammation required, the macrophages present in that space will be into M1 stage. And when the infection is cleared off and when the wound requires itself to be repaired, they will convert the macrophage into M2 stage, so that because they require ornithine for cellular and tissue repair. Is this clear?
Yes, sir.
Okay. Thank you. So we'll be moving to next slide. Okay, this is just a graphical summary of M1 and M2 macrophages. So if you can see here, when the macrophage is in M1 state, that is pro-inflammatory, it controls the tumor. It will kill the tumor cells. Right? But when the macrophage comes to M2 state, it will promote the formation of tumor. Promote in the sense the tumor cells convert M1 into M2 so that they can survive. The tumor cells can survive. Right? Okay. You might also see a word here, angiogenesis. How many of you can tell me the meaning of angiogenesis?
Sir, it is formation of blood vessel around like tumor cell or cancer cell.
Exactly. Not only tumors, any formation of blood vessel is angiogenesis, right? But there is extra angiogenesis in tumor. Why? Can anyone tell me? Though we'll be covering that in the cancer, hopefully, but just because I can see this word, can anyone tell me why do tumor as a mass of cell, they require extra blood vessel?
Sir, cancer cell or tumor cells needs extra glucose for like their faster rapid growth. So I believe because of that they need extra supply for through this extra blood vessels.
Exactly, exactly. Very correct. So see, the major route of transportation of oxygen and nutrition is the blood, right? Now, because formation of tumor requires extra cell division, and cell division is a physical process. That means the cells which are rapidly dividing, they require more oxygen and nutrition. And to get this more oxygen and nutrition, they require more blood supply, right? And for this
Yes,
extra
sir.
blood supply, they will form extra blood vessel around themselves for their own purpose. And that extra angiogenesis is is very important for a tumor to survive, right? And therefore you can see any tumor cell will always promote a macrophage in the M2 stage, not only for minimizing the inflammation, but also for formation of extra blood vessels. Is this clear?
Yes, sir.
Sure. Okay. You can also see these two terms, tissue remodeling and matrix deposition. We have just discussed in the previous slide. When the role of inflammation in an open wound is over, the cells in the wound will convert M1 into M2 macrophage. That means they will tell the macrophage that we no longer require nitric oxide, all the pathogens are killed. Now we require ornithine for our repair. And as soon as the inflammatory phase of the wound healing is stopped or it is over, the M1 macrophage in the wound area is converted to M2 macrophage, because now the wound require tissue remodeling for repair, and in case a scar is required, they will need extracellular matrix deposition. Is this clear? Any doubt to anyone?
Sir, can you just explain this matrix decomposition part just last
Okay.
briefly?
So the matrix here is extracellular matrix, ECM. How many of you have heard of ECM?
Yes, sir.
Right? It is the structure that supports the cell in a very layperson language, right? So when we're talking about a scar is the extra tissue. You can find a bump in the form of scar, right? That means extra amount of extracellular matrix is deposited to close the wound, right? And this extra deposition, even the normal deposition of matrix, is there because of M2 macrophages. Why? Because till the time macrophage is in the M1 state, it will keep on producing nitric oxide, right? It will keep on increasing the inflammation level. Now, a, a, a normal level of inflammation is very important to kill the pathogen. But in case the pro-inflammatory state remains high, how will the wound heal? Right? That means the wound requires M1 macrophage to just shut down its function, get converted to M2 so that the wound can get ornithine. And one of the function of ornithine, as we discussed in the last slide, is tissue repair. And how would a tissue repair without ECM? Is this clear?
Yes, sir.
Sure?
Yes.
Okay, great. Another very important aspect of ECM deposition is in tumors itself. All the tumor. See, how will you define a tumor, or how will you differentiate between a cancer and a tumor? Anyone? You might have heard uh, as a common sentence that uh, when the cells of the tumor goes to different location in the form of metastasis, we call that tumor as a cancerous tumor, right?
Yes, sir.
So though this definition is also correct, but again in literal sense, see, how many of you have heard of the term lung tumor or brain tumor,
Yes, sir. I
Yes,
have heard.
right?
Yes, sir.
Has any one of you heard the term blood tumor?
No, sir.
No,
Exactly.
sir.
So that's another difference, and that is one of the major literal difference. When we use the term cancer, we can use the term cancer both for the liquid tissue and the solid tissue. But when we are using the term tumor, it will always identify to a solid tissue, right? And that's why in every tumor, you will find a lot of extra ECM deposited, resulting finally to a state called fibrosis. And therefore, if there is a tumor formation in any part of the organ, okay, suppose we have a liver and there's a formation of tumor at this space, okay, this particular space will have extra ECM compared to other normal area. And because of the deposition of extra ECM or matrix deposition, which you can see here in the bottom, this particular tumor area is always harder than the rest of the normal tissue. And that hardness is because of fibrosis or deposition of extra ECM material. Is this clear?
Yes, sir.
Sure, everyone? Should I move ahead?
Yes, sir.
yes,
Okay, great. Okay, so this is just a image that where you can see different phases of phagocytosis. So if we can start from the left hand side top image. You can see a PAMP interacting with PRR, pathogen associated molecular pattern interacting with a pattern recognition receptor on the macrophage. I hope everyone can recall we just discussed about PAMP and PRR.
Yes.
Okay, as soon as these two interact, the whole complex is taken inside. Like this PRR is not like a gate that will open and the pathogen will go inside. The whole complex is taken inside in the form of a phagosome. You can see this phagosome. Now this phagosome interacts with lysosome. Fusion of phagosome and lysosome. That forms a phagolysosome. That means we have enzymes of the lysosome and we have the pathogens, which are broken down into fragments because of the enzyme. Now, once this is done, because if you can recall, we just discussed, phagolysosome is a temporary cellular structure. As soon as the pathogens are dead, the phagolysosome is dissolved and a new lysosome is generated. Now, as soon as the pathogen are destroyed into different fragments, these fragments, I will come to this MSC2 processing later on. This is antigenic presentation. These fragments bind to the newly formed MHC2 inside the cell. And this whole complex, the antigenic fragment along with MHC2, is now exposed on the cell membrane. You can see here. And that is what we discussed as the second function of macrophage, antigenic presentation. They present the fragment of the pathogen over their surface through MSC2 so that inactivated T cell, I'll use a different, so that an inactivated T cell can interact with this APC through T cell receptor. Now, when this inactivated or immature T cell interacts with peptide bound MSC2, it becomes T helper cell. You might recall MSC2 is responsible or interact with T helper cell while MSC1 interacts with T cytotoxic cell. We have been studying this, right?
Yes, sir.
Yes, sir.
Exactly. So when a phagocyte expresses, see here it is, when it expresses or exposes the antigenic fragment through MSC2 and the T cell receptor interacts, the T cell become T helper cell. And one of the function of T helper cell is to activate immature B cell into plasma B cell and memory B cell, so that plasma B cell can release antibodies into circulation. Is this image clear?
Yes.
Yes, sir.
Okay, great. Thank you. Now come to another cell, neutrophil. Okay, as we discussed earlier, neutrophils are the first cell that comes to the site of infection. They're not the first cell that respond to infection. The first cell that respond to infection are always the resident phagocytes. The first cells, the first immune cells that reach the site of infection are neutrophils. May or may not along with the monocytes, because a lot of macrophages are required at the site of infection. And that's why monocytes will definitely enter the tissue space to become macrophages. Okay, so neutrophils are the most abundant type of granulocyte that make up 40 to 70% of all white blood cells in the humans. So what do you mean by granulocytes? The cells that contains granules, and all these granules contains a lot of enzymes which are important to destroy the pathogens. Okay, so these granules have enzymes and other proteins that are crucial component of the innate immune system. That means a neutrophil will not differentiate the kind of pathogen, whether it's a bacteria, whatever kind of bacteria, or maybe a fungus or any other thing. If a neutrophil is activated, it will destroy the pathogen. On the other hand, agranulocytes don't have any granules in the cytoplasm. As you can see in the cytoplasm, there are a lot of granules. Granulocytes are also called polymorphonuclear cells because you can see the nucleus is not a single structure. It is multi-lobe nucleus, right? And this multi-lobe nucleus is a very significant characteristic of a neutrophil. Okay. Under homeostatic condition, neutrophils enter the circulation, migrate to the tissue where they complete their function and finally are eliminated by macrophages. So, when the neutrophil performs its function and the function is over and the neutrophil is about to die or is dead, they are destroyed by the macrophages itself. Because as we discussed earlier, it is the role of macrophage to clear off any dead cell. Now, when the neutrophil has performed its function, it will die its natural death, and all these dead neutrophils are also cleared by the macrophages. Neutrophils constantly patrol the organism for signs of microbial infection, and when found, these neutrophils quickly respond to trap and kill the invading pathogens. Now, there are three major roles or three major types of killing by neutrophil. A neutrophil is a very potent phagocyte. That means it can engulf the pathogen and kill, such as what is present in this particular slide. It can release the enzymes from these cytoplasmic granules. What you can see, these granules. Now, once the enzymes are released because of degranulation, these enzymes can directly kill the pathogen present outside. And the third mode of killing by neutrophil is release of nuclear material, that is release of the chromatid fiber or chromatin into a web like a structure. In Hindi, you might have heard of the term jal. Okay, so neutrophil also release the chromatin material as a web so that it can trap the pathogen, and this trap is called neutrophil extracellular traps or nets. And this process of releasing the nuclear material or chromatin fibers to trap the pathogen is called netosis. Because of the net, it is called netosis. Is this slide clear? Hello? Is this slide clear?
Sir, I have one
Yeah, please
question.
go ahead. Please go ahead.
So these cells are polymorphonuclear cells, as you mentioned.
Yeah.
I'm not sure. Is there a reason why they don't have a mononuclear like normal like other cells? Why they specifically have this?
Okay. So, see, they don't contain extra chromatin in their nucleus because the number of chromosome and number of genes has to be maintained, right? But see, if there will be a single nucleus, it may uh, it may acquire more space. But the cytoplasm of the neutrophils contains a lot of granules, which are highly dispersed, right? If you can see, just let me clear off this ink. If you can see, the lobes are connected through, just a minute. Yeah, the lobes are connected through this string. This is nothing but the chromatin material, chromatin fiber. Okay, to have a lot of these dispersed granules, the neutrophil has changed its single structure of nucleus into multi-lobe nucleus so that there can be proper dispersal of the granules. Plus, it will be easier for the neutrophil to release the chromatin material through any direction in case it want to kill the pathogen through neutrophil extracellular traps.
Okay.
Right?
Yes, sir.
Okay, just to give you one example, the fat cells or the adipocytes, okay, they are called fat cells because they have fat lipid deposition inside them as droplet. Okay, so sometimes there are multiple droplets of lipid inside adipocytes, sometimes there's a single large droplet of lipid inside the adipocyte. Now, because of the deposition of lipid inside the cell, the nucleus of adipocyte is almost every time pushed to the periphery, so that the inner part of the cell, intracellular space, can have more space to accommodate the lipid droplets. So that is one mechanism. So in every mature adipocyte, the nucleus will not be at the center as we normally know. The nucleus will be stretched or pushed to the periphery. Is this clear?
Yes, sir.
Okay. So neutrophils develop in bone marrow from hematopoietic stem cells in a process called granulopoiesis. That means granulopoiesis is formation of granulocytes. And mature neutrophils are characterized by their segmented nucleus and granules that are filled with more than 700 types of proteins in these granules. Granulocyte colony stimulating factor is the cytokine that finally converts the precursor neutrophil into a mature neutrophil. Within the eosinophil and the basophil, they form the class of with the eosinophil and basophil, neutrophil form the class of polymorphonuclear cells named for nucleus multi-lobed shape. The separate lobes are connected by chromatin. The nucleolus disappears as the neutrophil matures, which is something that happens only in few other type of nucleated cell. What is the role of nucleolus? Can anyone tell me? Nucleolus.
Ribosome synthesis.
Exactly. Very good. Now, because neutrophils reach a stage of such a maturation that they don't require any further protein synthesis, a mature neutrophil is at the end of its life. It doesn't require any further protein synthesis. And therefore, in a final mature neutrophil, there is no nucleolus. Because for protein synthesis, the cell requires ribosomes. Right?
Yes, sir.
Okay. In the cytoplasm, the Golgi apparatus is small, mitochondria and ribosomes are sparse, and rough endoplasmic reticulum is completely absent, so that more amount of granules can be accommodated. Another example, in a mature RBC, there is no nucleus, right?
Yes, sir.
Yes,
One
sir.
of the reasons for disappearance of nucleus is that mature RBC need to accommodate hemoglobin. Right? And therefore, there is no fresh protein synthesis in a mature RBC. Similarly, a mature neutrophil, because it does not require any fresh protein, it is at the end of its life, the Golgi apparatus becomes very small. You will hardly find any mitochondria, ribosome, and the endoplasmic reticulum is completely absent, along with the nucleolus. Is this clear?
Yes, sir.
Okay. Now, during the beginning of the phase of inflammation, particularly as a result of bacterial infection, because neutrophils are more active against bacterial infection. Okay. Particularly as a result of bacterial infection, environmental exposure, and some cancers, neutrophils are one of the first responders of inflammatory cells to migrate to the site of inflammation, as we have discussed. They are the first cells that reach the site of inflammation or infection. Neutrophils are removed by macrophages. Once the neutrophils perform their function and are dead, they are removed by macrophages and dendritic cells after neutrophils have phagocytosed and kill the pathogens. So these are the three types of killing mechanism of a neutrophil. They may release enzymes from their granules that will kill the bacteria. They may engulf the whole pathogen and destroy in the phagolysosome, or they may release the chromatin material and trap the pathogen. And now with this, once the pathogen is trapped, this whole structure is either taken inside, or this whole structure is killed by the macrophage. Is this clear?
Yes, sir.
Okay, great. Now come to the first mode of killing by neutrophil. It is phagocytosis. So prior to adherence to the target microorganism, the phagocyte or neutrophil begin to consume oxygen. It is very interesting part of neutrophil killing. Before it can engulf the pathogen, it consumes a lot of oxygen so that it can form a lot of reactive oxygen species or free radicals. This process of consumption of extra oxygen than it normally requires is called respiratory burst. Instead, the oxygen that is consumed to form reduced oxygen metabolites or reactive oxygen species, which will be used to kill the target microbe. The respiratory burst involves the activation of enzyme NADPH oxidase. Okay, just try to remember the sequence of the enzymes which are activated. The first enzyme that is activated for respiratory burst is NADPH oxidase, which produces large quantities of superoxide radical or superoxide molecule. This superoxide decays spontaneously or is broken down by enzymes known as superoxide dismutase. And human cell contains two isoforms of enzymes, copper and zinc, or along with manganese. Like these are the metallic cofactors in this enzyme, so superoxide dismutase. Either it contains copper and zinc, or it contains manganese. And this superoxide dismutase converts the superoxide into hydrogen peroxide. This hydrogen peroxide is converted to hypochlorous acid by another enzyme called myeloperoxidase. That means there are three enzymes responsible for production of final free radical that will kill the ingested pathogen. In sequence, it is NADPH oxidase, which produces superoxide from the oxygen, then superoxide dismutase, which produces hydrogen peroxide from the superoxide, and myeloperoxidase that will convert hydrogen peroxide into hypochlorous acid. And this hypochlorous acid will finally kill or destroy the pathogen that has been phagocytosed by the neutrophil. Respiratory burst requires a 10 to 20-fold increase in oxygen consumption through NADPH oxidase. NADPH is the key substrate of NOX2. The NOX2 enzyme in human is bound to the phagolysosome membrane. Post-bacterial phagocytosis, it is activated, producing superoxide radical. That means this NOX2, this whole complex of NOX2, of which NADPH oxidase is the primary member and NADPH is the primary substrate. This enzyme is bound to the membrane of the phagolysosome. And once the bacteria is inside the phagolysosome, the enzyme is activated and start producing superoxide radicals through the oxygen that has been consumed. Now, these superoxide radical will be converted to hydrogen peroxide, and these hydrogen peroxide will be converted to hypochlorous acid, and this hypochlorous acid will finally kill the pathogen. Okay. Peroxynitrite is a very strong oxidizing agent that can lead to lipid peroxidation, protein oxidation, protein nitration, which are responsible for its bactericidal effect. So not only reactive oxygen species, but reactive nitrogen species is also equally important part of neutrophil. Peroxynitrite is the most important reactive nitrogen species in the neutrophil that can cause lipid peroxidation. That means if there is a gram-negative bacteria, the lipopolysaccharide layer will be oxidized. That will result in disruption of the integrity and killing of the bacteria. It can also oxidize the protein of the pathogen. It can nitrate the protein, which has their own bactericidal effect. Okay. Any doubt till now? Is this clear?
Yes, sir.
Okay, great. Now the second mode of neutrophil-based killing is degranulation. It releases the enzymes present in the granules. Now, neutrophil has three types of granules: azurophilic or primary granules, specific or secondary granules, and tertiary granules. Azurophilic or primary granules are primarily loaded with antimicrobial protein called defensins. Azurophilic granules also contain myeloperoxidase, which is and also certain natural serine protease family. They also contain lysozyme and bactericidal or permeability increasing proteins. So these are the things present in the primary granules. On the other hand, the secondary granule or specific granule consists of alkaline phosphatase, lactoferrin, lysozyme, and NADPH oxidase, while the tertiary granule contains cathepsin, gelatinase, and collagenase. Amongst the broad ammunition that neutrophils carry in their granules, neutrophil serine protease directly kill microbe and inactivate the bacterial toxin. This particular enzyme is very important, neutrophil serine protease. Excessive host tissue damage and inflammation driven by the uncontrolled activity of these proteases, namely elastase, proteinase 3, and cathepsin G, is counterbalanced by endogenous serine protease inhibitors. Okay. See, the tertiary granules, as it is written here, contains cathepsin, gelatinase, and collagenase. Okay. Where in our body we find collagen? Anyone? Where do we find collagen in our body? Okay, not taking much of the time. Collagen is the primary component of any extracellular matrix of any organ. That means, what will collagenase do? It will degrade the collagen. Now, the degraded collagen is also called gelatin. Now, the tertiary granules also contain gelatinase. That means, if there is a activated neutrophil for very long time, what do neutrophil do? They are very potent cells that will destroy the bacteria. And if you can recall, we just discussed as example, any open wound is a great place for bacterial survival. That means, if the wound is needed to heal, there will be a lot of neutrophils coming to that area to destroy the pathogen, to destroy the bacteria. Even the neutrophil will destroy the bacteria because it contains elastase, proteinase, cathepsin, gelatinase, and collagenase. It will also destroy the extracellular matrix. That means, neutrophil is important for our wound. Suppose we take the example of wound to kill the bacteria, but because it has such potent enzymes, it will also destroy the natural tissue in the wound. That means, activated neutrophils for very long period of time will not allow the normal healing of wound. And that's why these proteins which are present in the tertiary granules of neutrophils are countered or counterbalanced by a lot of protease inhibitors in our body, which are, and the class is called serine protease inhibitors or serpins. That means, till the time we have active serpins in our body, these enzymes can, for no longer period of time, damage the tissue. They will be released. As soon as they release, they will kill the pathogen present in the periphery of the neutrophil and will be deactivated by the serpins. Because if the serpins don't work properly, these enzymes will be outside of neutrophil for a very long period of time and will start degrading the extracellular matrix and will not allow the wound to heal. When neutrophils sense the presence of microbes, they release these anti-macrophage.
Yes. नहीं
Yes,
बता
sir.
रहे कोई.
Sir, okay, sir. So see, if you can recall, or if this is new information for anyone present here, the bacterial, whether it's a cell wall or the LPS layer, they are anionic in nature. And when this BPI, when we are talking about this BPI, it is cationic nature. Suppose it would have been anionic, what would happen?
Repulsion.
Exactly. Like charges repel each other. And we have been studying since school. And that's why, being cationic in nature, they will stick to the bacterial cell wall or LPS and will increase the permeability so that the pathogen can be destroyed. And that is the importance of being cationic. And that is one reason. Okay, I'm just recalled of a very interesting example. There is currently, as per the discovery made till date, the only natural polymer that is cationic in nature is chitosan. Has anyone heard of chitosan? Chitosan. Some may also
Yes, sir.
yes. Okay. Chitosan is present naturally in fungal cell wall, and it is also a product of chitin. You might recall chitin is a very prominent structure present in the exoskeleton of arthropoda. So when
Yes, sir.
we break down chitin, we get chitosan. And chitosan, being the only naturally found cationic polymer, it is highly bactericidal. Okay, so I just recall from this example. So any doubt in this particular slide?
No, sir.
No? Okay.
No.
Okay. The last mode of killing by neutrophil is neutrophil extracellular traps or nets. And the process of release of net is called netosis. So in very simple language, what neutrophil does, it releases the chromatin material and traps the pathogen which is present on the periphery of the neutrophil. Okay. Now this net or the trap contain proteins from all the granules also. It's not just a physical trap that entraps the pathogen. When the neutrophil releases the chromatin material, it also along with the chromatin material releases the proteins from the granules, so that when the pathogen is now trapped and it cannot move, these proteins from the granules will do the rest of the work. It will kill the trapped pathogen. These proteins will kill the trapped pathogen. So nets or neutrophil extracellular traps provide for a high local concentration of these antimicrobial components and bind, disarm, and kill the microbes extracellularly independent of phagocytic uptake. Okay. Apart from just being a mechanism to kill the pathogen, nets may also serve as a physical barrier that prevent the further spread of pathogens. That means if the pathogen is present in the periphery of neutrophil, see, a pathogen which is not trapped, it can move to other location also. But if it is trapped with the nets, it will not move to other location. And therefore, this mechanism of sending out extracellular traps is not just to trap the pathogen so that the granule contents can kill the pathogen, but it also restricts the movement of the pathogen so that it cannot go to other location. Okay. The neutrophil extracellular traps might also have deleterious effect on the host also, because the extracellular exposure of histone complex could play a role during the development of autoimmune disease. That means, okay, we have just discussed in previous slides that about the damage associated molecular pattern, and we discussed about example of phosphatidylserine. That phosphatidylserine is a natural component and very prominent lipid present in the cell membrane. But in a live cell, in a normal cell, PS is present on the inner side of the cell membrane. And when a cell death occurs, PS is flipped onto the top. And the presence or exposure of phosphatidylserine is a damp for the macrophage, so that the macrophage can interact with the damp through PRR and destroy or engulf the damaged cell. Right? But in the process of sending chromatin material, so you may recall the very fundamental fact that chromosome is a structure that is made up of chromatin fiber and histone proteins, where the chromatin fiber is bound over the histone protein in a structure called nucleosome, right?
Yes, sir.
That means when
Yes,
the
sir.
neutrophil is sending the chromatin material outside in the form of net, because histone proteins are stuck to the chromatin material, they will be also coming outside the cell. And because immune cells, again, as I have said, immune cells are not trained to find histone proteins outside the cell. As soon as they will find histone protein outside the cell, they will start mounting immune response against the tissue itself. And therefore, presence of net for very long period of time is not good for the body, even though it is very good to kill the pathogen, but its longer presence is not good for the body, because the histone is now also out of the cell, and the other immune cell will recognize histone as non-self. Okay. Nets also play important role in inflammatory disease, such as nets could be identified in preeclampsia. Preeclampsia is a condition where pregnant women have continuous high blood pressure. Nets have also been reported in the colon mucosa of the patients with inflammatory bowel diseases, such as ulcerative colitis. Nets have also been associated with the production of IgG, anti-nuclear double-stranded antibodies, in children infected with Plasmodium falciparum malaria. Netosis has also been found to be associated with conditions such as rheumatoid arthritis, gout, thrombosis, and certain lung injuries such as cystic fibrosis. Because, as we have just discussed, presence of chromatin material or presence of histone particularly outside the cell is not a normal feature of the body, and therefore the immune system will start acting against the body itself, against the tissue, and may result in conditions such as autoimmune conditions such as rheumatoid arthritis, thrombosis, cystic fibrosis, ulcerative colitis, and start producing antibody against the double stranded DNA in Plasmodium falciparum infection. Is this slide clear? Any doubt? You may please ask.
Sir, I have one doubt,
Yeah.
that the net you mentioned is should be cleared by macrophages, right? So why there is like chances of prolonged exposure?
In case there is, let's see, we discussed earlier that macrophages patrol the tissues. That
Right.
means they come and go. In case, suppose, when netosis has occurred, there is very less number of macrophage around to clear the neutrophils. So suppose
Okay.
there are 100 neutrophils, and out of 100, 80 neutrophils are involved in netosis. But at that particular time, there are only 20 macrophages present around these 100 neutrophils. And this number of macrophages, suppose, is not enough to clear off the nets. That means the exposure time of net will be increased, right?
Yes.
Plus, as we discussed earlier, macrophages are very potent immunomodulators. That means they change their state from M1 to M2. And if they recognize net, there is very high chance that macrophage remain in the form of M1 macrophage, which produce pro-inflammatory cytokines, because these macrophages are seeing chromatin materials outside the cell for which they are not trained. They will always recognize these as non-self. So they will try to engulf the complete neutrophil along with the net. But in case they cannot engulf, such as if the number of macrophages is not sufficient, they will mount a pro-inflammatory response. And this excessive pro-inflammatory response may damage the tissue.
Okay.
Is this clear?
Yes, sir.
Okay. Now, I think this will be my last slide for today, hopefully. So, if you can recall, I've deliberately not put this picture that after that slide because it was just a term in that slide. But this is very important. You might recall we discussed about diapedesis. That means movement of immune cell to the tissue where they have been called to tackle the infection. Diapedesis. How many of you can recall?
Yes, sir.
Okay. So this is the figure of diapedesis. It is also called diapedesis is also called transendothelial migration. That means migration of immune cells across the endothelial membrane from here to here. And that's why transendothelial migration. So what happens? In a normal condition, the leukocytes are always rolling. They keep on rolling over the endothelial layer. As soon as they get a call from the cytokine and chemokine that there is an infection that you must reach immediately. Now, suppose these neutrophil keep on rolling, like this is a rolling neutrophil. If they don't respond to the signal or they don't receive the signal, they will keep on rolling. They will keep on changing their place. And from this place, they will move to this place because they are rolling over the surface. Am I right?
Yes, sir.
Right. Now, if they respond to the signal, the chemokine or cytokine released by the macrophage and the damaged epithelial tissue in the organ, in the tissue, upon receiving those signals, they start expressing a cell adhesion molecule called selectin. And not only neutrophils, the endothelial cells also start expressing selectins. There are different types of selectin we will study in the cell adhesion molecule chapter. There are P selectin, E selectin, and L selectin. L for leukocyte, P for platelet, and E for endothelium. So as soon as the neutrophil gets the signal from the cytokine and chemokine, it start expressing L selectin, and endothelial cell start expressing P or E selectin, and there is a binding between the L selectin and E selectin. You can see this binding. This binding stops the rolling of neutrophils. They serve as anchor. And once the rolling stops or halt for some time, neutrophil gets or utilizes that time to create a space between the endothelial cell so that it can move to the tissue space by changing their shape. And this movement of neutrophil from a rolling state to entering into the tissue is called diapedesis or transendothelial migration. And this is possible because neutrophil gets the signal through cytokines, or they act to the signal they receive through the cytokines. They start expressing L selectin, endothelial cell start expressing P selectin and E selectin, and the rolling stops for the moment because of the binding between L selectin and P selectin. And neutrophil utilizes this time of a stop to create a space between the endothelial cells and move to the tissue where it is required. Is this process clear to all?
Yes, sir.
Sure, everyone? Okay. So yeah, okay. This would be my last slide. So there are certain defects or diseases which are directly related to neutrophils. The first one, neutropenia, as you can see, is a very general term. Okay. For any reason, if there's a decrease in the count of neutrophils, that condition is called neutropenia. For any reason. So neutropenia can be divided into congenital and acquired. If there is some genetic defect right from the birth that is resulting in low neutrophil count, that is congenital neutropenia. And if there is a damage to the hematopoietic system and there is a low neutropenia in latter part of life, that is acquired neutropenia. One of the most common genetic defect that results in neutropenia is mutation in a gene called ELANE that codes for a protein neutrophil elastase. If there's a defect, because of any reason, if there is a defect or mutation in the ELAN gene that is not producing enough neutrophil elastase, there definitely will be neutropenia. It can also be a side effect of medication. There is a lot of medications such as cancer chemotherapy that temporarily decrease the neutrophil count, and that is the primary reason a, a person suffering from cancer receiving chemotherapy is highly susceptible to infection. You might have heard this, that
Yes,
cancer
sir.
patients
Yes, sir.
should be kept in an environment so that they cannot get infection. That is one of the prime reason. Though cancer itself destroys the immune system, but if the person is also on the top receiving chemotherapy, it is almost all most of the chemotherapy drugs are going to maintain a temporary neutropenia. Okay, so neutropenia is often combined with or comes with a fever, and that condition is called febrile neutropenia. Is a medical condition which is an emergency condition and requires broad spectrum antibiotics. See, fever is a symptom. Neutropenia is a condition. If there is a fever, that means the person, because of neutropenia, has got pathogens inside his or her body. And appearance of fever indicates that it is a medical emergency situation because the person is neutropenic. And that's why the person requires broad spectrum antibiotic. An absolute neutrophil count less than 200 is also considered a medical emergency. Even if there is no symptom, it is a medical emergency because the person may get severe infection at any point of time. And in those conditions also, even without any symptom, the person requires broad spectrum antibiotic if the count is less than 200. For a lot of conditions, neutropenia can be treated by a cytokine that comes as medicine, which is granulocyte colony stimulating factor, GCSF medicine. Another condition that is related to neutrophil defect is chronic granulomatosis disease. It is caused by mutation in the gene encoding the subunit of phagocyte NADPH oxidase. We studied in the phagocytosis mode of killing of neutrophil. NADPH oxidase is the most important enzyme that initiates the respiratory burst. And if there's a defect in this particular gene, and the incidence is around 1 in 2 lakh cases, this defect in NADPH oxidase gene result in a condition called chronic granulomatous disease. And most of the time, CGD will result in very severe staphylococcus or aspergillus infection. Another condition of neutrophil defect is leukocyte adhesion disease. If you may recall in the previous slide, the neutrophil requires adhesion to the endothelial cells so that it can find enough time to create a space and move to the tissue. If the neutrophil is not able to express selectin, even if the endothelial cell is expressing selectin, the neutrophil keep on rolling. And if the neutrophil is in continuous rolling condition, it will not move to the tissue space where it is required. Such condition where the neutrophil keeps on rolling and cannot adhere to the endothelial layer and therefore cannot move to the tissue space to tackle the infection result in a condition called leukocyte adhesion disease or LAD. And therefore, the infection remains as it is, the pathogen will keep on replicating and the infection will become more severe. Okay, this is my last slide for today. Any doubt in this?
No, sir.
No, sir.
Sure? Okay, so I'll try to stop the sharing. Okay. Now tell me the doubts.